Hello everyone, and welcome to this audio podcast. In this series, the Storm Focus v.3. This audio podcast will be in and around the sort of the main aspect within this series, and bring in a sort of an interplay into the overall theme of this audio podcast series, as well, if you like. So the audio podcast is here will be surrounding in and around chemokine receptors and leukocyte trafficking in acute allograft rejection, and aligned to observing chemokine and chemokine receptor expression in acute allograft rejection. This is a Ztorm Lipostics Vendip, and it's another Ztorm Lipostics production. So chemokine receptors and leukocyte trafficking in aligned to acute allograft rejection. So specifically in the early 2000s, research had been outlined, where research looked to assess the very early induction of chemokine and chemokine receptor expression in the immediate post-transplantation or transplant period. And the work allowed the research had alongside with the literature, I looked to understand how better immunosuppression and the maintenance of immunosuppression related therapy regimens and protocols within those who are suspect or at risk of acute allograft rejection would and could be at a more advantage if and wherein particular immunosuppression have been implemented, could be implemented, but also where at the same time to understand the basis in around how particular pathways in acute allograft rejection could be avoided if and wherein alternative immunosuppression regimens could be used or used in particular patients who were at risk of where there would be risk of acute allograft rejection, but also in a way to understand the trafficking of leukocytes, and understand chemokine and chemokine receptors. The research aimed also to observe donor-specific onset of acute allograft rejection episodes in turn also recognizing better allo responses and allo recognition. And this the research aimed to prompt a sense of increased prognosis to those to both the recipient and to both recipient and allografted and allograft post transplantation. So in context of transplantation, organ transplantation, the occurrence of one or more episodes of acute allograft rejection is a major determinant of allograft survival. Most episodes of acute allograft rejection are caused by cell-mediated processes and require the infiltration of allo-activated T cells in the engrafted organ. Over 40 chemokines and 20 inducible chemokine receptors have been identified. Um, In vitro investigations on the responses of activated T cells to inflammatory chemokines suggest substantial redundancy in this system, with multiple chemokines produced at tissue sites binding to multiple receptors on circulating leukocytes. There's works by Inston and Cockwell in the early 2000s that look at that have looked at chemokines and chemokine receptors. Chemokines binding to receptors on leukocytes mediate the sort of behind the scenes interplay between antigen presenting cells and host T cells in lymphoid tissues. The actual here and now, in your face fulfillment immune responses or immunobiology that can acutely destroy an allograft, and also, as seems increasingly apparent, smouldering inflammatory responses seen clinically as chronic rejection. Given these complexes and multiple roles, knowledge of chemokine biology is no longer for the enthusiast, and works by Hancock in early 2000 as well have highlighted the sort of biology in and around chemokines and chemokine receptors. The biological actions of chemokines are mediated through a family of G protein coupled receptors with seven transmembrane domains. The nomenclature used to describe these receptors is based on the class of chemokine ligands that interact with the receptor. So, for example, see the where the receptors are referred as CC, CXC, and CX3C receptors. Many chemokines bind to several different receptors, and the differential expression of the receptors by distinct leukocyte subsets is an important component of the specificity of chemokine action. The complexity and apparent redundancy on the system is thought to provide a high basis of effectiveness and flexibility in vivo. There's works by Segreger and team in 2000 that quite nicely highlight the works in and around chemokine and chemokine receptors and context of pharmacotherapies as well. Investigations with knockout murine models or knockout mice models, especially when teamed with the use in wild-type mice or murine laboratory models, blocking monoclonal antibodies to the corresponding receptors, have provided sort of the initial data to what all complexity what all the complexity of chemokine biology might mean to a highly practical in vivo world of organ transplantation. Recent investigations that have taken place between the early 2000s and a little prospectively, investigations have shown and highlighted that expression of multiple chemokine receptors allow navigation by leukocytes in a systemic fashion, although arrays of multiple chemokines so different through an array of multiple chemokines, so different chemokine receptor pairs may be necessary for activation and transmigration and retention accordingly. There have now been studies and investigations on the expression of a number of chemokines and chemokine receptors in human renal transplantation or organ transplantation that show increased expression in and around acute allograft rejection. Several important points have emerged so far from the initial in vivo based mechanistic investigations. First, targeting of a single chemokine is typically ineffective in prolonging allograft survival. An example of this general principle is the lack of efficacy in targeting of macrophage inflammatory protein 1 alpha rantes or of 1 alpha rantes, whereas targeting for receptors CCR1 and CCR5 is of pharmacotherapeutic significance. Second, chemokine receptors differ in importance as targets in allo responses. Thus, chemokine receptors such as CCR1 and CCR2 primarily promote macrophage recruitment to an allograft, such that targeting in knockout murine models, laboratory models, or mice have only a modest effect on allograft prognoses. And again, works by Hancock in the early 2000s have highlighted this, have highlighted context in regard to cardiac in around cardiac allografts. Third, the effects of containment immunosuppression or IST can modulate some of the rather black and white outcomes of chemokine receptor targeting in otherwise untreated laboratory murine or mice models. Thus, use of a subtherapeutic regimen of CSA or cyclosporin A has a synergistic advantage with targeting of chemokine receptor, which by itself has only a weak, for example, CCR1, or negligible, for example, CX3CR1 advantage. In the case of chemokine receptor, with the most impressive experimental data to align to role in acute allograft rejection, expression of CXCR3 and its ligands is closely associated with acute allograft rejection clinically. Works by Abdi and team provide data from 163 renal allografted recipients concerning the extent to which chemokine and chemokine receptor polymorphisms can affect the incidence of acute allograft rejection. The various stimulatory and aligned to this also the various stimulatory and inhibitory pathways involved in these activation cascades are providing insight into the design of potential for pharmacological agents. Segreger and team in the early 2000s informed that the immune system mounts distinct and selective immune responses to different types of infection and antigenic challenge. These have been termed Th1-like and Th2 type Th1-like and Th2-like after the two classes of T helper cells, T helper cells get involved. Th type 1 and Th2 responses appear to represent the extremes of a spectrum and aligned to immune responsive. Th1 responses are stimulated by pathogens that invade and inhibit cells and result in activation of cytotoxic T lymphocytes and delayed type hypersensitivity. The Th2 subsets produce cytokines and evoke strong antibody responses. So, for example, interleukin 3, interleukin 6, interleukin 10, and interleukin 13. A third subtype called Th0 secretes cytokines of both types, and it is believed to give rise to the polarized Th type 1 and Th type 2 lineages. Segreger and team in the early 2000s have highlighted have highlighted this, have highlighted context in line to this in their works. For CXCR3, virtually all T cells at sites of non-renal human inflammation with prominent mononuclear cell infiltrates suggest infiltrates are receptor positive, and works by Inston and Cockwell in 2002. 2000 in the early 2000 and 2002 highlight context in around CXCR3 as well. Growing evidence suggests that differential chemokine receptor expression may be crucial for the generation of a Th1 or Th2 type immune response, because specific chemokine receptor expression has been shown to characterize these T helper cell subtypes. Th1 cells appear to preferentially express the chemokine receptors CXCR3 and CCR5, whilst Th2 cells display CCR4, CCR8, and some CCR3. The chemokine receptors expressed by different populations of T cells may dictate to a significant degree where the tissue of infiltration of Th type 1 and Th type 2 cells, and the direction of the eventual immune response becomes more pivotal. In works by Cassieri and Springer in the early 2000s, they highlight quite nicely the chemokine receptor family. So wherein alongside there's chemokine receptors whose ligands have been identified are illustrated in a particular illustration in their works, where the chemokine receptor ligands are within a wheel of the illustration, and the chemokines ligands, the ligands are highlighted outside of the wheel as well. The principle of actions in and around chemokines are, for example, where they are released by stimulated cells and act locally. Chemokines are also produced by intrinsic cells as well as infiltrating cells. Chemokines act through G protein coupled 7-transmembrane receptors and reduce calcium influx. Chemokines also bind to proteoglycans and extracellular matrix. Chemokines induce hepatotaxis, where the cell migration along surface gradients. Chemokines also can activate adhesion molecules. Chemokines can induce oxygen burst and the release of granules and proteinases by leukocytes. And chemokines control aspects of immunomodulin, angiogenesis, hematopoiesis, and development. The process of leukocyte trafficking from peripheral circulation into tissue spaces involves a series of interactions between soluble mediators and surface molecules expressed by the endothelium and leukocyte, as well as subsequent interactions with extracellular matrix. Chemokines mediate events at multiple stages in this particular process. CXCR4 present on T cells, B cells, monocyte neutrophils, and blood-derived dendritic cells and others is most widely expressed. Is one of the most widely expressed chemokine receptors. CXCR1 and CXCR2 are expressed on most leukocytes, but appear to be fundamentally significant only for neutrophils, monocytes, macrophages, and mast cells. CXCR3, CXCR5, and CXCR6 are expressed exclusively on cells of a lymphoid lineage. CCR1, CCR2, CCR4 to CCR10 are expressed mainly on lymphocytes, monocytes, and monocyte-derived dendritic cells. CCR3 has multiple unique expression pattern, as it is found on eosinophils, mast cells, basophils, and Th2 type lymphocytes, and certain dendritic cell populations. Works by Olson and Ley in the early 2000 highlight more in and around this as well. CXCR3 is induced and maintained by interleukin-2, but inhibited by activation through the CD3 complex, whereas CCR3 expression requires the synergistic effects of interleukin-2 and interleukin-4. CCR5 is upregulated by Th1 cytokines, but can be expressed by interleukin-10. For T cells, activation through T cell receptor stimulation triggers a decrease in expression of CCR1, CCR2, CCR3, and CCR5. Increased expression of CCR7, CCR8, and CXCR5, and experimental condition-specific changes in levels of CXCR3, CCR4, and CXCR4. So aligned to this, 7 secondary lymph nodes, lymph organs play critical roles in the initiation of immune responses by serving as sites where naive T cells and B cells become activated through interactions with circulating memory cells and maturing dendritic cells arriving from sites of inflammation. Human immature dendritic cells derived from monocytes express the inflammatory chemokine receptors CXCR1, CCR1, CCR2, and CCR5, which allow these cells to follow chemotactic gradients to inflammatory sites. Maturing dendritic cells express large amounts of MIP-1 alpha and MIP-1 beta through 3 hour post induction, along with IP10, MCP2, and RANTES in a more sustained fashion. One consequence of this increased chemokine expression is the downregulation of inflammatory chemokine receptors on maturing dendritic cells, particularly CCR1 and CCR5, by homologous receptor desensitization. Although transcriptional regulation may also be involved, post-transcriptional events appear to dominate. The second consequence of this upregulation of chemokines is that it strengthens the original chemotactic gradient, further boosting recruitment of immature dendritic cells, monocytes, and T cells, and creating a continuous antigen sampling loop. In addition, maturing dendritic cells increase CCR4, CXCR4, and CCR7 expression and ELC and SLC responsiveness in a manner that is resistant to homologous receptor desensitization. In addition, maturing dendritic cells increase CCR4, CXCR4, and CCR7 expression and ELC and SLC responsiveness in a manner that is resistant to homologous receptor desensitization. CCR7 induces dendritic cell migration to secondary lymphatic organs. ELC, TARC, MDC, and DCCK1 are all upregulated by maturing dendritic cells, leading to increased interactions with and stimulation of T cells. With T cell zones of lymph nodes and Peyer's patches, activation leads to the production of polarized sets of T cells. Th1 cells selectively express high levels of CXCR5 and CXCR3, although functional CXCR3 is also expressed at low levels of Th2 cells. These receptors, along with decreased levels of CCR7, are thought to allow migration of Th1 cells to inflammatory sites, where interferon gamma has induced the production of IP10, MIG, and MIP1 beta. And again, works by Olson and Ley highlight some further context in and around this. The varied expression of chemokines within an allograft brings into question the best approach to targeting chemokines for anti-rejection in line to anti-rejection therapy. It is anti-rejection pharmacotherapy or pharmacotherapeutics. It is appreciated that it would be better to block early innate responses to an allograft, thus preventing the infiltration of non-allo-Ag specific on that will later recruit allo-Ag specific cells, or to manipulate regulation of later expressed chemokines. Thus, the question is also the question then is is there a pathway selectively inhibiting the adaptive immune response? So it is essential to block. It is essential to potentially block. However, further, it's important to another question is, which chemokines are the most crucial for in and around allograft infiltration, and must all parts of the immune response must all parts of the immune response be blocked, or is neutralization of only a specific few sufficient pathways or part of pathways encouraged to promote allograft prognoses? So, as observed in the cardiac organ, and of where gallium nitrate treated hosts, where gallium nitrate treated hosts are concerned, some chemokines persist even after allograft even after graft acceptance, with no apparent determinant to with no apparent detriment to the allograft. Works by Coven and Thompson in the early 2000s highlight an example in and around this. It was Hancock and team that used CXCL10 knockout laboratory murine models or mice models as knockout mice on murine laboratory models, wherein the mice heart were used as allograft donors or recipients, and found that donor hearts not expressing CXCL10 exhibited prolonged allograft survival, although recipients lacking CXCL10 had rejected in wild type murine laboratory or mice models at the same time. And so this suggests the data at that time that suggested that the ability of the allograft to secrete T cell recruiting inflammatory chemokines may be difficult, may directly result in its own acute allograft rejection. Other research using CCR1, for example, that binds both CCL3 and CCL4, sorry, CCL3 and CCL5, or CXCR3, which binds both CXCL9 and CXCL10, in around works that involve laboratory murine mouse models and knockout mice in around cardiac allografted in around research aligned to cardiac allograft recipients achieved some prolongation of allograft prognoses in the absence of anti-rejection therapy, but infinite allograft survival prognoses in combination with IST referred to as that has been referred to as CSA or cyclosporin A. On another note, CXCL1 anti-serum was given intravenously to mice or murine laboratory cardiac allografted murine mice cardiac allografted models, where the recipients 30 minutes post transplant disrupted the normal chemo where there was evidence that 30 minutes post transplant disrupted the normal chemokine cascade, resulting in a decrease in CXCL9 and CXCL10 expression, decreased cellular infiltrate, and also delayed rejection. Conversely, CX3 CR1 knockout laboratory murine or mice models were treated with a subtherapeutic dose of cyclosporin A dose of cyclosporin A and accepted cardiac allografts have been identified in to be where prognosis was longer than in CX3 CR1 wild type murine or mouse laboratory investigations. Works by Instant and Cockwell in 2000 or early 2000 in 2000 or 2002 to specifically highlight chemokines and chemokine receptors identified in human acute allograft rejection, where the chemokines in their works where chemokines are listed, the findings in around specific chemokines are highlighted, and there are some further citations in their work. It was Hancock and team who postulated targeting CXCR3 markedly decreases the tempo and severity of acute allograft rejection in vivo. The effect is associated significantly in decreased intragraft accumulation of activated T cells producing interferon gamma and conquest impairment of chemokine-dependent requirement of multiple effector cell types, suggesting a rationale of CXCR3 in synergy with conventional IST immunosuppression in clinical treatment and potentially in the management of acute allograft rejection. During renal diseases, the infiltration of monocytes, macrophages, and T cells into kidneys into renal pathophysiology is thought to play a central role in progressive interstitial fibroses and the progression of chronic kidney disease, or what was then referred to as, or has been referred to in literature as, chronic renal failure. And during this process, a crosstalk between cytokines, vasoactive substances, chemokines, and respective target cells takes place. The interaction contributes to the outcome, for example, healing or progression of the renal disorder. Chemokines appear to be integral players in this complex and dynamic process. Laboratory rat models in line to transplantation or organ transplantation have allowed a functional analysis of the role of chemokines in acute and chronic rejection. In acute renal renal transplant, an acute renal in acute allograft rejection. In acute allograft rejection, there is in acute allograft rejection in the laboratory rat models, that works by Nagano and team found that rantes and mRNA was upregulated by 6 hours during the initial organ transplant rejection phase, and after, and again post 3 to 6 days. Works by Nango retrospectively had been highlighted, have been documented or published in the late 90s. The functional role of rantes as its receptors was recently evaluated in laboratory rat models of acute allograft rejection using the rantes receptors antagonist metrantes. Treatment with metrantes significantly reduced the vascular and tubular damage associated with acute rejection and suppressed a mononuclear infiltration. Treatment with metrantes and low-dose cyclosporin A markedly highlighted markedly attenuated markedly attenuation where there was damage to vessels, tubules, and interstitial rejection had been evident. The potential clinical advantage of chemokine antagonists such as metrantes may lie in early effects on the suppression of cellular infiltration and subsequent allograft damage, which may help lower the inclination towards the development of chronic transplant dysfunction. Works have highlighted also that a chemokine receptor CXCR6 that is similar in humans and in murine or mice models has been identified to be induced on in vitro T cells primed in the presence of interleukin-2 but not interleukin-4. In blood, some CD4 positive and CD8 positive cells are CXCR6 positive. Very few CXCR6 positive cells are found in lymph nodes, but large numbers are seen or identified in sites of inflammation. The evidence suggests that CXCR6 may be a marker of effector Th type 1 and T cells, TC1 cells. CXCR6 binds the chemokine CXCL16, a transmembrane or a soluble protein produced by dendritic cells that attracts T cells and natural killer cells or NK cells. Works by Olsen and Ley highlight this in the early 2000s. In works that have been published in the early 2000s. Because of the goal of therapeutic intervention is disease modification and minimal impact on the normal host defense, the specificity offered by chemokines make them extremely attractive targets. It has previously been mentioned that survival of heterotrophic heart or cardiac allografts is prolonged in CCR1 mice. And that lower doses of CSA are required to completely protect allograft in these murine or mice laboratory models. It has now been established that a similar phenotype is observed in. It has been identified and established that a similar phenotype is observed in both CCR2 and CCR5 mice or laboratory murine mice models. In addition, antibodies to CCR5 attenuate the development of graft versus host disease in a murine model, although it is not clear if this effect is due to neutralization of CCR5 depletion of CCR5 expressing cells. Works by Cesari and Springer in the early 2000s highlight this. Schwartz and Radek in the early 2000s also found in TH type 1 dependent mice on laboratory murine mice models that immune nephritis mice or murine laboratory models had had a sequence of invading inflammatory cells that might be initiated by antigen-specific T helper lymphocytes and encountering their what would be referred to as their antigen in line to renal pathophysiology, which subsequently directs monocytes and possibly dendritic cells towards the injured tissue. In accordance with data, several investigations in human experimental glomerulonephritis or GN have highlighted that the expression of Th type 1 related chemokine receptors CCR1, CCR2, and CCR5 in the glomerular and mainly interstitial areas, as well as the urinary secretion of MCP type 1 is closely associated to disease activity and phases of progression. Again, works by Schwartz and Radek in the early 2000s highlight some further aspects in and around this. Cockwell and team in the early 2000s had shown that activated CD3 positive lymphocytes, which express the chemokine receptors CCR5, CXCR3, and CX3CR1, are highly responsive to chemoattractive factors produced by PTEC, which in turn have been stimulated by pro-inflammatory cytokines. Other investigations have shown a requirement for both T cell receptor interplay and the ligation of high affinity interleukin 2 receptor for full T cell activation. Cockwell and team also have demonstrated in blocking investigations, blocking studies or blocking investigations that chemotaxis of these cells is mainly attainable to ligation of CCR5 and CXCR3, and ligation of CX3CR1 has an additional effect. As works by Quinn in the late 90s had highlighted and identified that all activated T cells that express CCR5 are also CXCR3 positive, indicating that these cells may have chemo attracted through either receptor. Quinn and team also demonstrated that virtually all T cells at non-renal human inflammation having sites of non-renal human inflammation or where tissue aligned to tissue injury having prominent mononuclear cell infiltrates were CXCR3 positive. Works by Lotscher and team in the late 90s had identified that up to 40% of freshly isolated T cell express CXCR3, but these cells only migrate towards IP10 and CXCL10 after activation. In Cockwell's works, investigation, just over 20% of freshly isolated T cells express CXCR3 and 15% express CCR5, but no activation by anti-CD3 antibody, and approximately 90% of cells express both receptors. Differential expression of chemokine receptors coordinates specific leukocyte trafficking, which is important for the development, distribution, and deployment of the immune system. In contrast to lymphocytes, neutrophils and eosinophils more uniformly express smaller subgroups of chemokine receptors. Chemokine receptors have been interrogated. The interleukin 8 or IL-8 receptors CXCR1 and CXCR2 are strongly expressed on human neutrophils, and CCR3 is characteristic of human eosinophils. CX3CR1 is usually among chemokine receptors. Is unusual among chemokine receptors because it's not expressed on laboratory mice or murine models in context of T cells, but is expressed on 5% to 30% of murine or mice model natural killer cells or NK cells, and the work by Barlick and team in the early 2000s that highlight more in surrounding that in this. CX3CR1, like all chemokine receptors, is a 7 transmembrane domain G protein coupled receptor, or GPCR. A fractalene, so for example, CX3CR1 is its only known ligand, and CX3CR1 one is fractalene's only known mammalian receptor, and is classified amongst the inflammatory subgroup of chemokine receptors because it is selectively expressed on effector leukocytes. In addition to its expression pattern, it is unusual compared with other chemokine receptors in two main ways. First, it is one of the few inflammatory chemokine receptors that has only one ligand. Second, unlike most other chemokine receptors, it functions as an adhesion molecule. The precise biological role of CX3CR1 has not yet been delineated, or had not been delineated. However, CX3CR1 knockout mice or murine laboratory models highlight and have highlighted that there is a reduced susceptibility to specific complications present where there's presence of subtherapeutic doses of CSA, and where there's resistance to cardiac, and where there's also resistance to cardiac allograft rejection. Regulation of CX3CR1 expression has not been previously defined. However, because of interleukin 15 where it's essential for NK cell natural killer cell development and maintenance of in vivo, because CX3CR1 is expressed at high levels on a major subset of freshly isolated human natural killer cells, works by Barlick and team in the early 2000s hypothesized that interleukin 15 may be a positive regulator. Interleukin 15 upregulates expression of CC, but not CXC chemokine receptors on human T cells, but its effects on natural killer cell chemokine receptors, natural killer cell chemokine receptors, and on CX3CR1 on any cell type had not been reported in depth. Barlick and team or co-workers also tested the effects of interleukin 2 and interleukin 15 expression, and and also to do this, they developed a cytokine-dependent primary laboratory murine or mouse bone marrow derived bone marrow derived NK lateral killer cell culture system in which stroma was excluded. In the presence of interleukin-2 alone, for 10 days, the cultures consisted mainly of T cells, with only a small portion of NK T cells and NK cells. Interleukin-2 was able to stimulate selective outgrowth of particular NK1 cells if the freshly isolated bone marrow cells were initially cultured in interleukin-15. There are no available antibodies that specifically had specifically recognized mouse or murine CX3CR1 by flow cytometry. Barnick and team also discuss and conclude, although the results where the results have been documented to an extent, there have been some highly unexpected sort of findings for three main reasons. So three main bases. First, interleukin-15 is an essential factor for the development of NK cells, which, when freshly harvested from the blood, may express CX3CR1 in up to 30% of cells. Second, no chemokine receptor tested in any cell type had been reported to downregulate interleukin-15. And finally, interleukin-2, which is similar to interleukin-15 in structure and receptor uses, does not or did not suppress or has not suppressed or did not again suppress but instead modestly enhanced CX3CR1 expression. And again, works by Barnick and team in the early 2000s have highlighted have highlighted some works into some depth in around this. In works by Meltzer and colleagues who investigated expression of chemokine receptor CXCR3 and its ligand IP10 during human cardiac allograft rejection, they set out to investigate 169 sequential human endomyocardial biopsies by immunocytochemistry for infiltration by CD3 positive T cells and the representation of receptors CCR1, CCR3, CCR5, and CXCR3. The results identified and highlighted in association with CD3 positive T cell infiltrates and correlated with histopathological diagnosis of acute allograft rejection. The chemokines eotaxin, IP10, lymphotactin, MCP1, MIG, arantes, and SDF1 were analyzed in supplementary biopsies by real-time PCR. Eotaxin, IP10, lymphotactin, MCP1, MIG, and SDF1 were present in both normal and rejecting biopsies. Through CXCR3 ligands, IP10, which was rarely represented in natural biopsies, was markedly included in acute allograft rejection. Meltzer and associates and colleagues concluded in their works that the presence of CXCR3 positive T cells and the CXCR3 ligand IP10 within endomyocardial biopsies is concretely associated with acute allograft rejection. It was Hancock and team described in works CCR5 and its three main ligands IP IP MIP1 alpha, MIP1 beta, and RANTES, and the second most important pathway for rejection of vascularized allograft post CXCR3. The lack of host CCR5 expression causes a tripling of allograft prognoses in untreated recipients, and identified marked synergy with cyclosporin A or CSA and sirolimus, with CSA and or sirolimus. Analogous effects can also be observed or identified in wild-type murine or mouse laboratory wild-type mice laboratory or murine laboratory models through use of anti-CCR5 monoclonal antibodies. And again, works by Hancock in the early 2000s have highlighted some works in around this. Hancock and co-workers also highlighted the expression in around CCR5 and how CCR5 and how CCR5 contributes to the local recruitment of Th type 1 cells, and at least a proportion of macrophages during acute allograft rejection. And it is worth taking note of the data that had been occurring from analysis of co-stimulation where it involves substantial modulation of chemokine pathways. They still the team then have allowed considerable leukocyte recruitment to occur, and they are often antagonized by concomitant immunosuppression, antagonized by concomitant immunosuppression, in contrast to chemokine receptor targeting. Moreover, although blockade of co-stimulation with cytolytic T lymphocyte associated antigen perfusion protein or CTLA4A1G or monoclonal antibody to CD154 prolongs allograft prognosis, chronic rejection follows, which suggests that additional co-stimulatory pathways are still or have been identified or been investigated have been investigated in vivo. And aligned to this as well, in Hancock's and team's research, findings suggest that at least some of the co-stimulation blockade can be of therapeutic significance in conjunction with IST, and may act at least in part by modulation of early chemokine-dependent allo-activation. So, for example, they may act through suppression of interferon gamma and other cytokine production. This is supported by the similar outcomes achieved despite the manifold approaches used, and also the data described also related to the power of directly targeting CXCR3 and CCR5 chemokine receptor pathways. However, whether such modulation is in fact crucial to co-stimulation blockade, or is alternatively an epiphenomenon, needs to be further explored and identified. However, if CXCR3 was found to be expressed at some point post-transplant by a population of host regulatory cells that use this pathway to infiltrate a graft and maintain a local unresponsive state, then permanent suppression of CXCR3 expression, which could be counterproductive to attempts to achieve tolerance. In contrast to data and analysis in line to the roles of chemokine pathways in acute allograft rejection, exploration of the role of chemokines mediation of chronic rejection. Chronic rejection is only been initiated or been investigated since the early 2000s, at least in context of chemokine receptors, in line to observing chemokine and chemokine receptor expression, progress in investigation of chemokines in chronic rejection has been slow, and in large part because of the unsatisfactory nature of laboratory rodent models of allograft rejection. And should one use a weak so for example, should one use a weak non-MHC disparate models such as the favored what have been evidenced as the favored Fisher-Lewis combination in laboratory rat models, and argue that events therein have some relevance to the clinical chronic rejection, or should one use more vigorous laboratory models with full MHC differences, suppress acute allograft rejection, and hope that some form of chronic rejection takes place thereafter. And again, Hancock and team highlight some aspects in some depth in their works in the early 2000s. The particular significance of the laboratory rat or the rat Fisher-Lewis model is that it allows the use of orthotropic renal allografts, i.e. so for example, both host kidneys are removed and a transplant is placed in the normal location, together with the surgical repair of renal vasculature, ureteric drainage, and direct effects of chemokines on the recruitment, proliferation, and activation of vascular smooth muscle and other non-hemopoietic cells, in addition to mediation of leukocyte recruitment, adds to the challenge of the situation. And in the soul renal investigations, induction of CXCR3 and IP10 mRNA has been associated with acute allograft rejection. And so finally, in a clinical representation by Hancock and team in the early 2000s, T cells within a procedure referred to as bronchoalveolar lavage of lung transplant recipients undergoing rejection episodes were CXCR3 positive and exhibited a strong in vitro chemotactic response to IP10. Graft infiltrating macrophages expressed IP10, and alveolar macrophages expressed and secreted IP10, and levels capable of inducing chemotaxis of CXCR3 positive T cell line. IP10 production was upregulated by interferon gamma, and high levels of CXCR3 ligands were verified with bronchoalveolar lavage fluids of recipients with rejection who were aligned to having acute allograft rejection episodes. Once again, more the research in around the context of this can be identified in works by Hancock and team in the early 2000s, and again works by Inston and Cockwell in the early 2000s can also be identified in their works. So this draws this audio podcast to a close. This has been an audio podcast in a line to Chemokine receptors and leukocyte trafficking and acute allograft rejection. This has been an audio podcast in and around the Ztorm focus v.3. This has been an audio podcast aligned to the series a series also aligned in and around and aligned to observing chemokine and chemokine receptor expression in acute allograft rejection. It's a Ztorm Lobostics Vendip, and once again this has been another Ztorm Lobostics production. Thank you.